So my goal today is to provide you with an introduction to cancer and really answer the question of what is cancer. And to do that, I'll provide you with a little bit of information on DNA and the genes that are critical for the development of cancer, as well as information on the types of cancer, cancer staging, and cancer treatment. But I think if you ask most scientists and oncologists what cancer is, they would indicate that cancer is a defect of the cell cycle, particularly with respect to ongoing replication or proliferation. So in this schematic, a normal cell represented in pink converts into a cancer cell. And as you all know, cancer very much rapidly divides to ultimately produce a tumor. But at the heart of that is defects in the cell cycle. And the cell cycle is really a process by which a cell goes through to prepare itself to be able to divide. And there are certain checkpoints in that have to be maintained and are actually lost in cancer. Just a little bit of information on some of the DNA. As you all know, chromosomes are the structural units of DNA. And in humans, there are 23 pairs or a total of 46 chromosomes in every human cell. And in fact, if we took that DNA from those chromosomes and unwound it and laid it end to end, that would equate to approximately 2 meters of DNA per human cell. That's the equivalent of approximately 40 kilometers of a very thin thread thrust into something the size of a tennis ball or a nucleus. But not only is it pushed into that nucleus, but rather it has to be organized in a manner that allows certain genes to be accessed and expressed and other ones not expressed. So each human has approximately 2 times 10 to the 11th kilometers or 200 billion kilometers of DNA. That's roughly the equivalent of 66 round trips to the sun and back. Now, this is an extraordinarily thin thread, and the weight of which is only 300 grams, or roughly 300 paper clips. So it's not difficult to envision how defects in the genome or this DNA blueprint can really adversely impact cell biology and contribute to cancer development. Now, here is an image of a microscopy technique where we're able to independently label all 23 pairs of chromosomes. And you can see that this is a normal individual or a normal cell. But in cancer, that normal balance is very much disrupted. Here we can see what's called numerical changes in chromosome numbers. So rather than the 46 copies that you would find in most human cells, we find 127 copies. So each chromosome is massively amplified in this a single example of a breast cancer. But this can be applied to many different cancer types. Similarly, we can also see structural variants. So while we see the regular number of 46 chromosomes, what you'll notice in many of these chromosomes are banding patterns, indicating fusion proteins and fusions have occurred between these specific chromosomes. Really, there are 2 classical genes that are implicated and affected by those types of numerical or structural changes. The first one is oncogenes, and these are really referred to as the cellular gas pedal. It's as if a car really has those accelerators stuck. As a result, the genes are turned on and the proteins have increased expression and function and really are driving cells around that cell cycle. The second genes, which really keep those oncogenes in check under normal conditions, are referred to as tumor suppressor genes. And in a car analogy, these are really referred to as cellular brakes. And in fact, in most cancers, you lose both of the cellular brakes, both your pedal brakes as well as your emergency brakes. So these genes are turned off, and as a result, they are often decreased in expression or function in many different cancer types. Now, moving back to those cell cycle, there are a number of stop signs, or also referred to as checkpoints, that normally prevent the cell from prematurely progressing through the cell cycle to ensure that it's ready to move through and ultimately divide. And so those genes very much play roles in those. Under normal conditions, those oncogenes are pushing the cells through the cell cycle, but those tumor suppressor genes are acting as those brakes at those specific checkpoints to allow DNA repair genes to normally come in and repair the genome. But if we lose those tumor suppressor genes or those brakes, those cells will go through the cell cycle and accrue many mutations that may ultimately contribute to the development of cancer. In general, there are 2 cancer types. On the left are liquid or hematological cancers. These are broken down into 2 types. First and foremost is leukemia, which arises through blood-forming tissues or cells, and an example is acute myelogenous leukemia. A second type is lymphoma, which again arises through blood cells, but of a specific type, those being lymphocytes. And an example here would be Hodgkin lymphoma or Burkitt lymphoma. Now, the other type of cancer is a solid cancer, and this again gets broken down into two types. Carcinomas, which you may be most familiar with, these arise through defects in epithelial cells or linings of organs. And these can include breast, colon, lung, and liver cancers. Alternatively, we can have sarcomas, which basically are defects in the supportive tissues and can include bone, cartilage, or muscle. And an example here is osteosarcoma or bone cancer or fibrosarcoma. Now, in general, there are more than 200 distinct types and subtypes of cancer. In fact, it's estimated there's at least 18 distinct breast cancer subtypes. And it's really this diversity or heterogeneity that makes it so challenging to combat these specific cancers. Another term you may have heard of is cancer staging. And this is a very important aspect of cancer biology because it provides the medical team with important information and insight into the patient's cancer. It provides information on location, cell type, tumor size, and spread, or whether or not the disease has metastasized throughout the body. So in this schematic in the middle, it's a single example from colorectal cancer where we have a cross-section of the bowel, and I'm just showing you the different stages from stage 1, effectively up to stage 4. Now, stage 1 and 2 are referred to as early stage, and that is because this disease is locally restricted. And that generally means that the disease remains in place where it originally started. Stage 3 and 4 are late stage or more advanced stages, and these are usually stages at which the disease has begun to spread or metastasize throughout the body. And in fact, in stage 4 shown here is colorectal cancer often metastasizes to other organs, like in this case, the liver. Now, tumor staging is very important because it provides that critical information that's used to develop those treatment strategies. But what are the factors that impact treatment decisions? First and foremost, the tumor type, whether or not it's liquid versus solid. Tumor staging, whether or not it's locally confined or begun to spread throughout the body. And again, patient characteristics such as their age, health, obesity, or other factors. In general, there's two types of treatment strategies. One is a local therapy, typically for those early stages when the disease is restricted to where it originally started. And those include surgery and radiation. And the second real treatment strategy is systemic treatments for more of those late stage diseases. And this typically involves chemotherapy. Now, I hope today I've provided you with a little bit of information and an introduction to cancer biology and begun to answer the question of what really is cancer. And with that, I thank you very much for your attention.